Welcome to Neurosalience, the OHBM podcast.
Welcome to the OHBM Neurosalience podcast. I'm your host, Peter Bannattini, and I interview neuroscientists, brain imaging, and modeling experts from around the world to discuss their work as well as challenges of using imaging to understand the brain and to help those in a clinical setting. Today, it's my pleasure to have Dr. Ahmed Khalil as the guest. Dr. Khalil is a MD PhD. He's currently serving his residency in radiology at the Institute of Neuroradiology at Charité University Hospital in Berlin. Before starting his residency, he was doing pioneering work towards developing the use of resting state bold latency mapping to reveal flow deficits in the brain associated with stroke. This approach compares very well with the current gold standard approach of dynamic susceptibility mapping of an exogenous contrast agent, such as gadolinium, that's used clinically worldwide. He's written several papers on this bold latency approach for looking at flow deficits that include the methodology, applications, comparisons with gadolinium, and also comparisons with, for instance, the use of DTI for looking at where the lesions are, stroke lesions. And it all compares very well. The technique actually can get useful data in about 2 minutes, as he's shown in one of his papers. So the technique is great, and we have a really good discussion, not only about this method, but also about just the wider issues of how to move a method that seems like it's useful clinically into clinical practice. What would it take to move this method into daily clinical practice on individual patients around the world, on scanners around the world? So we talk a little bit about that. We talk a little bit about the advantages and disadvantages of the method. We talk about his own struggles, as with all MD PhDs, sort of balancing research and clinical practice. And we also talk a little bit about the future of artificial intelligence in radiology. And we bring that up in the context of even how to best use this technique and how maybe AI might help bridge the gap between the cultural sort of standards of clinicians just looking at grayscale images or certain other types of data that are minimally processed. So that's one problem with fMRI, is actually to sort of package the processing in such a way that minimizes artifacts and increases the interpretability and the digestibility by a radiologist. But if you could have AI help out, that'd be great as well. So it's a great conversation. Dr. Khalil is inspiring. He has a lot of energy. He gives great advice, and has a very humble approach to, assessing this method. And I really do think it has a lot of promise. So I hope you enjoy this conversation. Okay, welcome to the OHBM Neurosalience podcast. I'm Peter Bannattini, and today I'm with Ahmed Khalil. Ahmed, he has an MD PhD. He's a resident at the Institute of Neuroradiology at the Charité University in Berlin. And he's been pioneering with collaborators at Max Planck and also at Charité, a technique that I think is the lowest hanging fruit for actually having fMRI-like things instituted clinically. And that's looking at resting state bold latency variation over the brain to identify areas of flow deficits, either with stroke or other things. And so it's a nice technique. We'll talk about that towards the end of the podcast, but also Ahmed is very active, or has been. Now he's just busy doing his residency, but he has been active with his blog, which has a lot of really good thoughts on how science is done, how things are published. And we'll talk about that maybe starting out and go from there. And I just wanted to also, last thing, mention that I first knew of Ahmed because he did something I think that more people should do. So I had written a blog on clinical applications of fMRI, and I didn't really mention this technique, but he actually went ahead and emailed me and said, hey, this is a technique that should be maybe potentially mentioned, and caught my attention. And I think it's always good to put yourself out there and reach out and assert yourself wherever you can, and because good things happen. So it caught my attention. He's had my attention ever since, and I'm doing everything I can to try to promote this as well. So from there, Welcome, Ahmed.
Thank you so much, Peter. Thanks for the invitation as well.
Yeah.
Very happy to be here.
Well, it's great. It's great to have you. So just to start out, I always start the podcast with a little bit of the history of the person being interviewed, and basically, you're an MD PhD. And so, what first piqued your interest in medicine or radiology or even the brain?
Yeah. So I think it started when I was in high school. Basically, I kind of realized that the subject that interested me in the most in high school was biology. So I was like, okay, I'm interested in biology. What can you do with that? And initially, actually, I was thinking more about going into something like molecular biology or something like that. And so I went to high school. I'm from Sudan originally, and I went to high school there. There weren't so many programs for things like molecular biology or anything like that at the university level. And I was like, okay, so what's the other thing I could do? Medicine sounds interesting. My older sister had studied medicine before me, and so I kind of had a little bit of an idea about what it was about. And I thought, okay, great, that sounds like something that I could envision myself doing. And then my interest in radiology actually came much later, but initially I wanted to become a neurologist in medical school. I found just neurology very, very fascinating. And then after I graduated from medical school, I had some postgraduate kind of exposure to neurology, and it was really what I expected. I just realized that the kind of day-to-day of it wasn't really for me. And at the same time, I was thinking, you know what, I'd done a little bit of research during medical school, and then I did some research after graduating as well, before starting my internship, basically the first year of residency or the equivalent in the US. And I thought, okay, I want to do some research. So how can I get into that? Some more research. And I thought, okay, I'll apply to master's programs. And I finally got a scholarship to study here in Germany, master's in medical neurosciences. Two things kind of maybe primed me to like a career in radiology. One thing was in medical school, like we had of course all these clinical rounds in different specialties and so on, but the rounds in radiology really I found were really interesting for, I think, a very specific reason. Like we had a professor of radiology, and he would, during the rounds, just be sitting there in his room, in his reporting room, and he was just this really interesting character. He had this like impeccable suit on and a bow tie, while all the other, I don't know, attendings were running around in these like shabby white coats and stuff like that. And he just had, I just thought, this guy's he's like sitting there, he's doing such interesting things, and his character really left kind of an impression on me. Yeah. And so that was one of the things I think that kind of primed me maybe to a career in radiology.
Yeah, it's interesting how like little things like that, like just seeing how a person holds themselves and kind of seeing their whole essence and sort of thinking, okay, I want to be, then it's all builds around that. It's so much information in somehow that sort of thing. So you said you came from Sudan, and did you? I mean, I imagine, is the program all in English or in German or?
So the master's program was in English. It was an international program. It was actually this European scholarship program. It used to be called Erasmus Mundus. Now it's called Erasmus Plus. So it basically allows you to pursue postgraduate studies, masters or PhDs in at least 2 different European countries. So I chose Berlin here in Germany and Bordeaux in France. So it was all in English, basically. So
okay.
Yeah.
And so then how did you start in the program did you first start just saying, oh, I want to collaborate with these people who are doing this? Like, I don't know if it was Arno Willinger or maybe his group, or did you want to do that thing and then you sort of picked them? Like, how did that work?
Yeah, the collaboration with Arno and the Max Planck came a little bit later, but actually while I was doing my master's studies, basically we were supposed to do a couple of what they called lab rotations, so short research stays in different research groups. So you're kind of left, like you basically have to find a lab and apply there and kind of stay with them for 4 to 6 weeks, I think it was, or something like that. And one of the people who taught us our neuroanatomy classes in the master's program is now a very good friend and colleague of mine. And after she taught us one of the lectures, I just kind of approached her because she had spoken about like the research that she was doing and so on. And I was like, yeah, it sounds really interesting. Medical imaging, MRI, with stroke patients. Do you have an opening for a lab rotation? And she said yes. And so I joined, working initially on just kind of standard perfusion imaging, DSC, MRI. small project. And then I continued basically in one of the points of the lab rotation is that you can potentially continue for your master's thesis in that same lab if you want to, if it fits. And I ended up continuing working on then fMRI-based perfusion imaging. And as I was kind of wrapping up the master's thesis project, that's when the contact with Arno and Arno Fillinger and the MPI came about, because of course Arno's back then a former PhD student was one of the people who basically first showed that this fMRI-based perfusion imaging works in stroke patients.
Yeah. Okay. Yeah, that's great. And sort of and jumped right into it. So before we talk a lot about that, we'll talk much more about that. I just, as I was doing research, I like to research the work and everything like that. I came across your blog, and you brought up some really interesting points, and I just wanted to maybe talk about those a little bit. I mean, so you're right at the interface of, you're doing clinical work, but you're also doing methods development. Or you have been, and you will be. And there's always this strange relationship between that. I mean, it seems like it's such a cumbersome, not really cumbersome, but long and drawn out sort of process by which methods get implemented clinically. But then also, if you do clinical trials, sometimes, as you wrote in your blog, sometimes the clinical trials show a positive result, but then later another study shows it not a positive result. And so we're sort of kind of mired in this sense of what works, what doesn't work. And of course, there's also the issue of publishing. A lot of people publish, and they maybe not as much with clinical trials, but like with their latest method or whatever, and they show that it works, but they don't really reveal all their cards in terms of, what they're concerned about, because they want to sell the method. And sometimes that goes too far. But science eventually just corrects itself, but it takes a long time. So what is your thought on the process of methods development to clinical application?
One thing that I think it's a very fundamental issue, but I think it's something that's really important. It's about kind of how methods research generally is perceived by the greater scientific community, especially maybe by clinicians, people who do clinical work or clinical research. And what I really think is that when it comes to methods development and validation, I think you have to start with earlier, like low-hanging fruit. I think you have to start with those things. I mean, I sometimes tell my students, some of my favorite projects that I've worked on started with, I don't know, or not started with, but when I would explain maybe at the beginning what I'm trying to do to someone, kind of maybe feel them internally rolling their eyes a little bit, thinking, well, that's obvious, or why are you looking at something so basic, where's all the exciting, cutting edge, groundbreaking stuff. And I'm not saying that those things aren't important, but I'm just saying that the very basic methods validation, answering these questions that are seemingly very obvious, I think it's just extremely important. My response there is always, it might seem obvious, but there's a lot of things that seem obvious that don't work, that kind of make sense logically or whatever, but end up actually completely failing. And also this idea that, for example, it's really simple, like, why are you looking at this, or very, very kind of obvious. It's always like, okay, but if it's so obvious, why isn't there any empirical evidence on it, right? So you just want to generate that empirical evidence, or why is the empirical evidence so weak and so, I don't know, maybe a poor quality or something. So I think that's in my opinion where it all starts. I think in general, as a scientific community, we have to rethink how we perceive such research, because of course there's absolutely a place for this, daring kind of really kind of cutting edge research questions and so on, but it all has to start somewhere. And so I think that's a really important part of it.
Yeah, exactly. Like a lot of the clinical questions are much more, are very different than, the clinical metrics are different than the research, what people want to do with, research. You just want to find new things and show something is really cool. It kind of works. But with clinical, it seems like you want to, establish its efficacy for patient treatment, for diagnosis and treatment. And it has to be very solid. And
Exactly.
the solidness is it's kind of like, the researchers would be like, oh, that's sort of a mundane sort of question of, whether, you have to do like X number of patients and sort of compare it and carefully with the current techniques. And it seems like sort of like that it's not the most flashy sort of thing, but it's what is necessary for clinical implementation. And yeah. And I don't even know, I know that's, and it's funny too, because some clinicians, it's interesting because my brother's a psychiatrist and, he's very up on sort of the latest trials on drugs and things like that. But then at the same time, he'll sort of wing it sometimes and sort of just use his intuition and try something or, nothing too strange, but it's sort of like based on his own like intuition about what kind of would work. And he just tries things. And so it's like, there's this really, it's kind of a disconnect between, there's this established research, but then still the clinicians just, try things.
Yeah, absolutely. And I think that's really important. Like both of those things are very important, and this kind of maybe experience, intuition based, or kind of implicit experience based theories and things that lead to research questions and so on, that they're also very important. And also on the matter of like this issue of, do you do this kind of mundane kind of methodological research, or do you go for something bigger? I think we're big and bold. I think we're going to speak about this maybe later when it comes to statistics and so on. But I'm also very much convinced that from a statistical research methodology standpoint, kind of extraordinary claims require extraordinary evidence, right? So you need a different statistical approach. You need more rigor if you're dealing with these kind of hypotheses, these research questions that I'm not saying are not correct or anything, but they're just a priori, there's less evidence for, or they just kind of seem a bit more far-fetched.
Yeah. No, I see what you're saying. So I have I was thinking along those lines when I saw your blog post on that. I have a interesting take. Statistics. I think that's really important. I also worry a little bit about people doing sometimes. Every statistical analysis requires some sort of model of what you're looking for, and I often worry that sometimes statistical tests can be perfectly correct, but still miss or be incorrect in the sense that you could have an artifact that shows up as statistically significant because it sort of follows that, and you don't model it out, and so it's always, you never should totally rely on, you should always kind of, it's an art to sort of use the statistics well, but then be able to back off and say, oh, is this really the right thing we're capturing as far as that's concerned?
Yeah. And I think that's related to something also like, when you're a clinician doing methods research, I think you're quite sensitized to the fact that in the end, your overarching goal in most cases is something to do with an individual patient, like one person, right?
Yep.
And bridging that gap between statistics, group statistics, and individual patients is extremely difficult. And there's no real even like framework or anything for doing it. And that's why I think something that is related to this, but also related to this issue of maybe lack of translation or translation failure between basic methods research to clinical practice, is the fact that especially in those early stages when you're mostly validating and developing methods, I always think this kind of radical transparency approach is the best. So do your group statistics, make sure everything is as rigorous as possible. Of course, we all try to do that, but at the same time, whenever possible, show all your work, right? And that involves, a lot of times that involves, showing individual level results. That might be, I try to do this in my own publications, you upload individual level data to some repository, link it there, and maybe nobody ever takes a look at it, but whoever's interested in taking a look at it can go through those results one by one and see how they at least translate it at this stage to the individual level.
Yeah. You really hit it on the head, and I really think that this is I think people are still grappling with this problem. The whole idea of doing group studies and translating a group study to an individual patient. And that's what I always say with a lot, there was a paper that came out, several years ago on BWAS, brain-wide associations. And they, it was very kind of a depressing paper, but it was a really good paper saying, look, with fMRI, resting state, you need 2,000 subjects to derive this difference. But I always worried that it might be, so I think that in group studies, there's two problems. Yeah, you don't know how it translates to individuals, but also sometimes if you don't group your populations well enough, you could average out or properly do the analysis, taking into account every individual difference. You could average out the effect, and it looks like it's no effect, but in fact you have to be more selective in your patient population, and sort of that's also kind of an art in some sense. And it would be nice. One thing that would be nice is like each clinician trying something. It would be nice to actually have their, of course, you have case studies, and that's a publication but it would be nice to somehow have worldwide databases of what individual clinicians' experiences are with a technique, and so they can follow up. Because usually all that data is lost. They try to treat the patient, and then whatever happens, and then they don't report that. It's not like into a repository in some sense. Yeah, a couple of times, right? I would ask a physician, okay, what are the odds that this will work or not? And they don't really know, or else they have different criteria for success or not, and so it's very murky. If they could capture that individual clinician experience, it would help.
And I mean, there's precedent for that. For pharmaceuticals, that's what you do, right? After you've done all your phase 1 to 3 studies and the drug is out there and licensed and being used in clinical practice, there's this continuous kind of feedback loop. Okay, sometimes it works well, sometimes it doesn't work so well, but there is a framework for doing this, right? For monitoring side effects that might have not come up in the initial studies, or efficacy differences between individuals, that kind of thing.
Yeah, so that's good. That's good. And it would be nice to capture that even more with other things too. But yeah, no, that's perfect. I mean, that's so important too, because there's clinicians' experiences. That's the real world. And I would love to have fMRI sort of like things used on a day-to-day basis clinically. And I don't think enough people are thinking hard about how to. I mean, we're still sort of caught up in the research, and that's a thing that now gets you promoted and whatever, but not as much on these sort of infrastructure to sort of narrow it down to individual subjects. And so I think the first paper that I saw that came out where you were first author, where you first introduced the idea of, I have a JCB-FM, stroke 2017, and then JCB-FM in 2018. It's 2 papers for looking at bold latency, resting state latency, and comparing it with current techniques for looking at vascular patency here. And so could you talk about how you got into that and what it's all about and what you did?
Yeah, so that started, so this was my master's thesis project in 2014, but actually the story starts a little bit earlier, because in 2013, there was this paper by Liv et al., also with Arno, I think is the last author. And they showed essentially for the first time that using fMRI, you can detect deficits in perfusion in the brain in patients with ischemic stroke, an acute ischemic stroke. And this paper itself was based on actually ideas that have been brought up by Lays Frederick and Yunji Tong, because they were looking basically at these oscillations they were looking at oscillations. I think at the very beginning, they were using pulse oximetry and NIRS, near-infrared spectroscopy. And they found essentially that you have these oscillations, these traveling waves that essentially go throughout the body and including the brain. And you can track them in a way that, in a relatively simple kind of way. And you can show that these kind of move throughout the body and in a way that looks very much like vasculature, like vascular movement, blood flow. And then Yating Liv, who was a PhD student of Arno's, essentially in this 2013 paper showed that you could use that to show these perfusion deficits in stroke patients. And then 2014 is when I kind of took over that project, because basically Arno is of course at the Max Planck Institute in Leipzig, but the data for that Liv paper was collected here in Berlin at the Center for Stroke Research in Berlin, where I was doing where I did my lab rotation and then my master's thesis. And so the idea was to then take over that project and see, first of all, can we validate these results in a larger cohort? Because the first paper was kind of a proof of principle, I think, with 15 patients or something. And we had collected in that time up until 2014 a lot more data. And that paper actually also compared resting state-based perfusion to DSC MRI, dynamic susceptibility contrast MRI. And the idea was just to do the same thing essentially in a larger cohort with a bit more quantitative comparisons, correlations between the metrics and so on. And that became that Stroke 2017 paper. Yeah, and then basically in this larger cohort, we were basically able to replicate what editing Liv had found and also showed that this delay in the bold signal, so these delays in these oscillations that run throughout the brain from a single brain region compared to a reference signal, they correlate the most with delay-based parameters from DSC, so Tmax and TTP, time to maximum and time to peak. Yep.
So just to ask a couple of questions about that. So, people are probably thinking, oh, wait a second, we use resting state for looking at, connectivity. And so the oscillations is about like 0.2 hertz. And just to be clear, like this oscillation isn't like, so there's pressure waves that come with cardiac that's faster. There might be waves that might be correlated with breathing that might be somewhat linked to that. But these waves are sort of like the 0.2 hertz. So, and then basically I think that the main method that you did was you just took the overall average, the global brain signal, averaged that, used that as a reference function, and then calculated the delay on a voxel-wise basis relative to that reference function. And so that kind of captures the global signal, which we actually always try to regress out.
Yeah.
But that's sort of the useful signal. And so that delay, it's interesting how that oscillation, it sort of indicates sort of like oscillations in flow and oxygenation that occur really slowly and they kind of like, wash over the brain related to the flow. And then another thing too, like you said, you compared it to perfusion, and maybe you can talk about this, is that, I have a friend who's in neurology. He's a PhD, but he's trying to develop perfusion techniques. And I think he stopped doing that because as nice as perfusion is, it's very insensitive. It's very noisy. And he's like, when you have a patient in there, you have to get the information fast, and you can't just be averaging for like an hour to get a good perfusion signal. So, and bold is nice because it seems like this technique, has much higher signal to noise, and you can get the information more quickly. So my question is, is that still the common technique, using like a global signal as opposed to maybe like a signal off the photodiode on your finger? And then also my main question is, so you have these papers, you have a whole series of papers sort of talking about using it, all these comparisons and, case reports and looking at with MS and other things like that, and also comparing to diffusion imaging as well, which diffusion actually gets at edema. So it shows where the lesion actually is. So how does it compare with the gold standard, I guess, which would be gadolinium or some sort of dynamic susceptibility contrast to look at perfusion? How does it compare? Does it get you more information in some ways or less information? And, how do the maps compare? Are they clinically useful, and in how much time?
So to your first point, in terms of what we're looking at, like what these bold oscillations actually represent, it's, I think, largely still an open question. One thing that was not clear, I think, to most people, was definitely not clear to me at the very beginning, was whether it wasn't, let's say, simply the cardiac signal being aliased, because you have a relatively low temporal resolution, at least with standard fMRI acquisitions, 2-3 seconds or something. So you're aliasing all those cardiac respiratory signals into these low frequency range. I think that question has largely been settled. It's not that, because we now have multiband sequences with higher temporal resolution, and you can kind of get rid of most of the cardiac signal just simply by filtering and redo the analysis, and you still see very similar changes, the same changes. But of course, there's still a lot of things that it could be within those within that low frequency range. There's the classical hemodynamic response function, even at rest, of course, right? The neurovascular coupling. There's vasomotion, there's all kinds of things, there's Meyer waves, like all these things that oscillate at exactly, or more or less exactly, those frequencies, right?
Yeah.
So what exactly is driving it is still an open question. To the choice of reference, one thing that I think over the years has just surprised me the most is just how robust the technique is to different references. So Blaise Frederick and Yunji Tong, like I said, used essentially a reference from the finger, pulse oximetry. We've used a global signal, a single hemisphere, the venous sinuses, and it all works. They're different. The results are slightly different, but it all still gives you generally the same results most of the time. And we settled, at least in our group, we settled on the venous sinus reference. I think for me, for a couple of reasons, it showed slightly better correlations with Tmax and TTP in our stroke paper. Small differences, but they were still kind of there. Second of all, conceptually, it just kind of makes more sense to me. The venous sinuses are the outflow tract of the brain. There's no let's say, if neurovascular coupling is a contaminant, let's say a noise signal in this context, then you don't really have that problem in the venous sinuses, right? But it's also just kind of from a temporal aspect, it's cleaner. It's like the end, and so it's just easier to interpret, I think, than having a global signal averaged up. Yes, so that's the reference part. And then with the signal to noise issue, I mean, of course, generally fMRI doesn't have a great signal to noise ratio, right? Or at least when people usually think about it in terms of task fMRI, just like how in that sense, the signal to noise ratio is quite low, right? With something like DSC and gadolinium, you've got quite a high signal to noise ratio, right? The gadolinium really causes the big change in the signal. Of course, things like motion and so on, they contaminate your analysis and confound your analysis. But still, in terms of signal to noise, it's a pretty big change. On the other hand, fMRI-based perfusion, in my opinion, has the advantage of the signal being, I mean, it's pseudo-periodic, right? So it's not perfectly periodic, it's pseudo-periodic, but essentially it more or less kind of repeats itself over and over. So you don't, I mean, just kind of in a very simple way, you don't have to catch something, right? It's kind of constantly happening. And as long as you get a clean enough kind of segment of signal, you should be able to get a reliable analysis. And that's what we did to answer your question about like clinical practice and how long you would need to basically scan in order to get reliable results. That's what we did in this paper in Frontiers in Neurology, was it 2021, I think. And we just basically took this a 6-minute scan, which is really kind of the longest we could get in these acute stroke patients, because it's just a very difficult clinical population to keep in the scanner for too long. We took that 6-minute scan and we just kind of shortened it artificially, kind of, and looked at the effect of shortening the scan. And the conclusion from that was that about 2 minutes, in most cases, gets you pretty reliable from a clinical, again, diagnostic point of view. We had radiologists look at the images and tell us if they can see the perfusion deficits. And we also kind of quantified this by taking the full scan as a reference. About 2 minutes of data gets you quite reliable results, both from a quantitative perspective as well as a qualitative kind of clinical perspective. Of course, motion is an issue, as it kind of always is with fMRI, because motion is complicated and the changes due to motion are sometimes much larger than the changes like the background oscillations are, and spin history artifacts, and everything's very, So, but generally speaking, about 2 minutes of data does get reliable information.
And this is at 3 Tesla, and maybe standard resolution, like 2 millimeter voxel sizes or something like that. And,
exactly. I think ours is 3 millimeters, 3x3.
Oh, 3x3.
Yeah.
Okay.
Yeah.
And that's good enough. Yeah. I mean, I imagine that, right. Even if you go to higher field strength and whatever, and you might want to, so you could do maybe better, and so it's sort of like a quick asymptote of like, more is better, but, at some point it sort of plateaus and maybe that's around whatever, 5 minutes or something, but that's not really totally necessary to make the diagnosis. So.
Exactly. I mean, it would be interesting to find out where it plateaus, just kind of from a methodological perspective. In stroke patients, it's very hard to scan for 10 minutes or longer in acute stroke patients, at least. But yeah, it would be interesting to know, because for functional connectivity, let's say, there is some evidence for that, right? So people have scanned for kind of really, really long times, and kind of that plateau is more or less kind of been established, although I guess there's a bit of controversy there. But with bold-based hemodynamics, it's an open question.
Okay. So then my question is, so as far as the information content, I mean, it's still, dynamic susceptibility, you inject this big bolus of gadolinium, it causes the signal to decrease like 40%, and then you integrate onto the curve and you look at the latency of the curves and calculate perfusion and so you get a quantitative, people like it because they get a quantitative measure and they're more comfortable with that. But with bold, it's relatively qualitative, and it's sort of, you try to, since we don't know exactly what's causes the oscillations and exactly how this relates to perfusion, it seems like it lines up on the maps. But what are your latest thoughts on, the information content? And is there any risk of, seeing something that looks like a perfusion deficit that's not, or false positives or false negatives as far as the maps are concerned and how they compare?
Yeah, I mean, when you look at the individual maps, especially we started relatively recently to look at the individual maps in non-stroke patients, so patients with or people with vascular risk factors, hypertension, diabetes, and so on, but without acute pathology, at least not no acute brain pathology. Yeah, you do realize that there's a lot of variability in the, let's say, noisiness of the maps. It's really, to me at least, quite unclear where that variability is coming from. Is it an issue with reference? Is it something intrinsic? Is it motion? Is it intrinsic, I mean, to the message itself? Is it motion? It's very hard to tell, and that's also why these more established perfusion imaging methods, ASL and DSC and so on. I think maybe your colleague from neurology would agree. I mean, it's not that they don't have this noise or anything, but people who interpret them, radiologists and neurologists, they're just more used to seeing them, and the noise just kind of blends into the background. In DSC with Tmax or something around the ventricles and in the posterior parts of the brain, you always see these things that look like delays that are probably not even a method, like a technique related artifact, but something physiological to do with the posterior circulation dynamics or something. These kind of things. I think we just need to, and that's why looking at these maps, these bold lays in or people without any gross brain pathology, and obvious brain pathology, is very useful because it just helps us get a sense of what the range of normal is. Right?
But for the most part, I mean, it's so. Well, maybe to back up a little bit. So, how are these maps used? I mean, of course, it's nice to see where there's a flow deficit, and it's interesting. Like you have one paper comparing DTI versus with this to see, like, acutely is the tissue still viable, and so is it worthwhile to use methods for opening up the flow? Sometimes that causes problems, and so how does it inform treatment?
You have the diffusion lesion, which is generally considered to represent tissue that is no longer viable. Some controversy about that. It's not entirely true, but generally speaking, of tissue that's no longer viable. And then surrounding it, usually you have an area of perfusion deficit that you see on perfusion-based weighted or perfusion imaging. Part of that perfusion deficit, not all of it, represents the operationalized ischemic penumbra. So tissue that is at risk of dying, of becoming irreversibly damaged. But like you said, if certain treatments are applied to restore the flow, its function can be restored. And with DSC, one has to say it's also not a very clear-cut issue how to define the ischemic penumbra in on DSC. But there's empirical data on that. And people have used thresholds of, for example, Tmax to operationalize this ischemic penumbra on DSC. And that's what we tried to do with Boldley as well. And we did come up with a threshold that was fairly accurate at identifying this region of potentially salvageable tissue. But of course, as you alluded to, I mean, the true test of it would be to test it in practice in a clinical trial, right?
Yeah.
That's what you would need to do. And that hasn't been done yet for several reasons.
Yeah. Well, I mean, that's interesting. So actually maybe leading into that. And so before I lead into that, in terms of processing, so it seems that there was recently a paper, and I noticed that Daniel Margulies was on it as well, looking at, which I thought was kind of a nice idea. I mean, just applying ICA to this analysis. And so it seems like just looking at the papers, it seems like the maps are cleaner in some sense, even more clean when you do ICA of the whole time series and pull out these specific frequencies. Yeah. I mean, so are you moving to that as far as the analysis is concerned? That was a paper published in Human Brain Mapping in 2021. And it seems like it's nice because it helps to maybe further automate the analysis procedure. So could you describe maybe that a little bit? And
yeah, just for sort of context, I mean, just how we stumbled upon this finding. Basically, I just wanted to do ICA of fMRI, of the fMRI data, actually to look at effects of kind of denoising, removing head motion from the data, and see if that improves the bold delay maps. And then as you do with ICA, and around the ICA, and I had to kind of look through the individual components of individual patients, so not group ICA, but single subject ICA. And yeah, I was just kind of looking through the components, and then I just started noticing these components that don't look anything like noise to me, don't look anything like any resting state networks, but look a lot like the perfusion deficits that I knew these patients had because we had DSC to compare it to. And it was one of those things where, I mean, I didn't really, in the beginning, I couldn't really believe it. I looked through every single component of every single patient that we had. I think the entire cohort was like hundreds, a couple of hundred patients or something. But it's just showed up surprisingly consistently. And of course, like this is one of the things that kind of comes with maybe trying to do methods research as a clinician, is that if I were, for someone who's like more method, has more of a methods background, it makes complete sense. If that area of perfusion deficit has its own signal characteristics,
Yeah.
or at least fairly kind of different to the other components, then it's going to show up as a component, right? So, but anyways, so we noticed that you get these components really kind of consistently, and then we started looking at the implications of that. So first of all, you don't need a reference, right?
Yep.
So you get the reference is completely kind of, you've solved kind of that issue. Second of all, the big thing was head motion. So if ICA can separate components that are kind of due to head motion, does that mean that the remaining components, including this, what we call the hyperperfusion independent component, this HIC, does that mean it's cleaner? Like you said, at least visually, it seemed that way, cleaner than the corresponding bold delay map.
Yeah.
And of course, then there's the automation thing, is that, of course, ICA by itself, you don't have to give it too many inputs or anything. It just kind of gives you components. But of course, there's another aspect of the automation, which is, okay, you get all these components. How do you pick out the one that represents hypoperfusion? And that's something that we tried to address in this human brain mapping paper by using a relatively basic machine learning algorithm, basically just to we trained it on a subset of the data, and then saw how it would perform on identifying these hyperperfusion components in the remaining data. And it did quite well. And I think one of the reasons that it did quite well is because the underlying signal is so different. Even when you compare it, so for sure, comparing the noise components to signal components, that's known from fMRI research, they're very different. But comparing these hyperperfusion components to resting state networks was also in terms of their signal characteristics very different. You have lower frequencies, lower amplitudes, and a delay, and so they're time shifted compared to the resting state components or networks, and so that helped us kind of pick these out and automate the analysis even further. And in terms of which kind of, I still don't see them necessarily as kind of competing methods, like the implementation of the bold delay or of this fMRI-based hemodynamics that we have currently running in our research group actually runs both, because I think for me at least, it's still too early to decide for one or the other. Yeah.
Okay. So, all right. So then now coming back to clinical implementation. So how ready, this is sort of like a pretty robust and maybe useful. There's more contraindications with gadolinium. People are more sensitive, you're realizing that you don't always want to use it. It seems like it could be something that could catch on quickly, clinically. So, what would be the steps now? You'd have to do a clinical trial on this. And then I always worry about, radiologists are used to just looking at grade scale images and, adjusting the knobs, looking at the contrasts in their own way, and then making a decision. But here, as the problem with all of fMRI, it's like there's a lot of processing, not a lot, but there's some processing steps, and then the radiologist has to relearn how to look at these maps, or the neurologist has to figure out how to look at these maps and make an interpretation on them. So then you'd have to have like some, really solid, standardized processing suite that you just press a button and it comes up, which I find is still difficult. And then you have to disseminate it, everywhere, and and have them all try it. And so what do you think, so what's necessary and what do you think how do you see that happening? It'd be nice to get this in all the scanners all over the world. How would that be done?
I think um, I think just to preface my answer, I think the relearning how to look at the maps part, in my opinion, is the much bigger problem, because simply it'll take more time. There's no way around it. They have to expose radiologists and people looking at these maps to the maps. There's no other alternative. They've been used to, like you said, looking at DSC and all these other kind of MRI sequences for decades, and this is kind of completely new. So the processing part, there are of course a lot of different parameters that could potentially be tweaked here and there, but that is, in my opinion, also completely true for DSC. There is really, I mean, it seems kind of maybe. Seems that kind of, if you just kind of look at it, it seems like maybe DSC has kind of all figured out in terms of processing, but the algorithms, they vary. There's no standardized approach. They're just completely, and I'm not saying they don't work or that's a bad thing or anything, but it's just, there's a lot of variability.
Yes.
So I think the processing part, at some point in, I think it was 5 years ago or something, we decided, okay, there's a million different things you can tweak in the processing, but generally speaking, it seems like a robust method, relatively robust to these more or less kind of minor change or differences in processing parameters and so on. So we started focusing on usability. So we're just like, we're going to define some kind of, for us, standardized processing pipeline. We're going to implement it on our scanner, and the real work will be in just making it as user-friendly as possible. And so we started to do this, and we have it running on our scanner at the Charité here. And basically, it involves the clinician, the radiologist, kind of opening up a browser window, logging onto the server, and then with 2 clicks of a button, like just 2 clicks of the mouse, and waiting for 2 or 3 minutes, they have the maps in front of them. Okay. And what was important for me when implementing that as well was not just the usability, but this kind of error detection or kind of quality control type thing. So they not only get the maps, but they get a graph of motion. They get some other kind of parameters that hint towards how good the quality of the data is. And we have that implemented, and it's, I mean, we kind of made it ourselves. It's not like not a product kind of, it's not a, it has its flaws, let's say, but it's, but doing that taught me that actually the processing part is actually not that hard, because if you had 2 or 3 max programmers or something, they can make something in a week that's maybe 10 times better or something. So I think that the real problem with widespread implementation is awareness. The vast majority of people don't know that this method exists. The target audience, let's say, they just don't know that the method exists.
Yes.
And the second thing is experience with the actual maps.
Okay. Yeah, it seems that, right. I mean, as far as the DSC, I know, my wife is a cardiologist, and they do DSC at the heart, and they have their own, at the NIH, they have their own, custom processing suite, and they do stuff, and it works, and they like it. But you're right. I mean, that's sort of like becoming more prevalent, but it's still, and I think what happens is, I mean, you're working on a Siemens scanner, right?
Yeah.
Yeah. So often I think that what you have to do is get the vendor to buy into the idea. And if they see a potential clinical market, they'll be like, okay, we'll take your processing method, we'll make it into a package. We'll start selling it for like, whatever, $30,000 for each user with a research agreement. And then people start using it then. And then it sort of catches on. And then maybe simultaneously clinical trials are done. And then it's sort of like this iterative process in some sense. But you almost have to, and it's interesting too, because it can't be necessarily open source software, because it's nice. And this is actually something I have to remind myself, like, it's really great to have open source software so everyone can just have it for free. But to actually make it used clinically, it almost has to be, some company has to take it on towards the goal of making a profit or something. It has to be the package that you then invest in and make right and standardize.
Absolutely.
Yeah. So then, so you mentioned the cultural thing with radiologists sort of learning to do this. I I'm even thinking, and this might lead into the next part of our conversation, is at some point, maybe sooner than later, and you talk about this a little bit in your blog, is the whole use of AI. So maybe we don't even have to have the radiologist learn how to interpret this. We could just, AI algorithms will just pull it out and then give a suggestion to radiologists as to what to do or whatever. So what do you think of the future of AI in terms of even interpreting this or in general for radiology?
Yeah, I mean, I think it's a really important point because on the one hand, I mean, we're dealing with such a complex signal, right?
Yeah.
And I mean, it's a complex signal. It's there's a lot of just a kind of mind-numbingly large amount of physiological processes and so on that go into this, that are reflected in this one signal. And of course, that allows us kind of, we can take that same signal and we can use it for functional connectivity. We can use it for all kinds of aspects of hemodynamics. I mean, I'm focusing on mostly latency and so on, but there's people, using fMRI for CVR measurements and all kinds of different things, vessel imaging, large vessel imaging, all these kind of things. So that by itself seems like it would be quite useful for some kind of AI-based implementation because it is because that's kind of what AI is best at doing, right? Taking these complex signals and that are either they're hard to interpret generally, but also maybe one of our weaknesses kind of as humans is that we take this complex signal and we just kind of break it down almost arbitrarily into this is functional connectivity, that is CVR, that is perfusion. So I think there's definitely some potential there. I still have no kind of concrete idea of how that would actually work, but it just seems like a method where AI would be quite useful.
Yeah, they can get, once they get enough learning sets. My feeling, I mean, I don't, it's not really based on much, but just sort of an intuition that I feel that the information that you can get from looking at this sort of global resting state signal, the latency and maybe other aspects of it as it correlates with other things like respiration might be more informative in some different ways than just using gadolinium. So it's hard to say right now, but and maybe AI can help take in all this data and pull that sort of information out potentially with the right guidance. I mean, there's limits to how much people can iterate. That's one of the strengths of AI is that you can gather in a lot of data and consider a lot of variables at once and sort of put it in this parameter space, figure things out. But so yeah, who knows? I mean, maybe this might. I mean, I think it's a useful technique. I think there's more information there even to get out. And one other idea. So this is a random, as I was looking at your papers, a big thing at ISMRM, and even everyone's trying to do glymphatic imaging because it's sort of like maybe an early indicator of Alzheimer's or even related to other psychiatric diseases or whatever. And it's sort of maybe related to brain inflammation in general. It almost seems that instead of looking at bold, you could maybe sensitize your sequence to CSF. Just looking at CSF, maybe long echo time, a little diffusion weighting, something like that. And then redo this analysis, just looking at CSF to see, delays and flow and things like that. And that could also be an interesting technique. I don't know if you've thought about that. I mean, maybe there's papers in the literature on that, but
I mean, that's a really interesting point because there's I mentioned earlier that the source or the sources of these oscillations that are driving these the source of these systemic oscillations are kind of unknown. But one of the things that is potentially responsible or kind of have been postulated as being potentially responsible is exactly that CSF flow, lymphatic flow. And I know like Yunji Tong, for example, has been looking at this, I think, with looking at inflow of CSF right at the base of the brain and using that as a reference signal and trying to essentially visualize CSF flow in that kind of way. And of course, CSF flow and lymphatic flow are very closely related.
Yeah.
So there's definitely some potential there. And I think that generally speaking, the tricky thing about doing that, in my opinion, is that there's no reference standards, right? Like at least for perfusion, you have this, albeit it might be imperfect, but you have DSC, you have other things that are relatively well established that you can use to compare it to.
Yeah.
While with lymphatic flow and CSF flow, I mean, there are things, phase contrast, MRI, and so on, that they work in kind of very different ways, and making those comparisons, I think, is really a challenge. Of course,
Yeah.
that's where preclinical animal studies might be very useful, because there are more invasive ways to look at CSF flow or lymphatic flow, and that could be. But the problem is, I mean, I worked on this a little bit during my PhD, trying to establish this fold delay in a mouse model. It was extremely difficult. Like, I don't know what it is. I don't know if they don't have these systemic low frequency oscillations. I don't know if it's something to do with the sequence that we use.
Huh. Yeah, the limits of preclinical, maybe the vasculature is organized differently. And it seems like my feeling is like such a small brain, it's sort of, more efficient in everything getting there at the same time. And interesting. So, okay, well, yeah, we're coming up in an hour, but I just want to have a few just, final questions and points. So as far as what you're doing right now, I mean, right now you're mostly in residency, and so you're bogged down in clinical work, but as far as like maybe what you plan or hope to do once you get out of residency, try to establish a lab. I always am so impressed with MD PhDs, because the ones who are successful seem like they're doing like two jobs at once. I mean, they're doing clinical work, they're doing research, and they're somehow making it all work. Do you plan on doing that sort of thing? And then what excites you most? Do you want to continue this work, or what are your thoughts on that?
Yeah, I mean, I go back and forth about, like, with this in my head a lot, because I have to say, I was never good at this, like, really kind of doing these two things at once. So that's why I always had either a kind of full-time research position, or at the very least, something like 20% research or something, where I could just have like real dedicated time to doing research. But so I don't really know what I kind of plan to do after residency in terms of my research, and also, yeah, establishing, like, trying maybe to establish a lab or something. I'm not quite sure if it's really for me. I mean, I don't know if we want to get into that, but it's like, I just to kind of, I think one of the things that I enjoy the most about research, when I really think about it, I mean, I enjoy several aspects of it, like writing, science communication, and presenting work and so on. But it's really like, I don't know, sitting with my colleagues and students for 2 hours or something in a room, and just kind of brainstorming and trying to figure out solutions to like complex problems and so on. And I don't know too many group leaders or anything who have kind of the luxury of being able to do that, at least on a regular basis.
Yeah.
So I'm still a little bit undecided on whether that's something I'd like to pursue. But in terms of like, if I kind of go back to at least a substantial amount of research or something after residency, I mean, the fMRI-based hemodynamics, I think, is something that I'd like to continue to pursue. And what I would kind of like to focus on would be this idea of integrating different aspects of different types of information that you can get from fMRI together, right? So if you take just one fMRI scan, and how much information can you get out of it? Functional connectivity, perfusion, CVR, vessel imaging, and whether kind of using that all together leads to any kind of, or has any kind of clinical relevance.
Yeah. Well, I think you get a lot of traction with that. Now, of course, right, the MD PhDs they grow teams of people to sort of help them. But I think actually, I think that would be a great thing to do. I think it would actually be useful for the field too. And not only for the field, but for ultimately helping patients. And yeah, I do think that there's information in those oscillations that's still untapped and clinically useful. I also, just to reiterate, I think that as far as clinical implementation is concerned, this has more, I think, promise. I mean, everything has promise to some degree, but this has more promise than even the bold activation stuff in many ways. So as far as a couple of last questions, but as far as the skills that you had to learn, your background was more biology, but I mean, did you have to learn, the right computer science or processing or things like that to do this as well? So.
Yeah, I had to. I mean, I had, the fact that I spent 4 years on my PhD full time, and that was, for me, it was great. And this just kind of goes back to the point that I think for me, it wouldn't have worked like doing part-time research work and clinical work at the same time, because I don't think I would have been able to dedicate so much time to learning. And I had to learn a lot of things. I had to learn signal processing and programming, and really also the technical methodological side of medical imaging was also, I mean, that's, at least I didn't learn that in medical school, kind of at a really just the absolute basics of like CT, MRI, that kind of thing. So I had to learn a lot of that. And but in that sense, I think having this consolation during my PhD of working in a medical imaging department at the Charité, having a lot of like methodological support from people at the Max Planck Institute. And then my third supervisor was also here at the Charité. He's a physicist, Philipp Böhm-Sturm. So having those kind of three things together, and one day I'd be here and one day I'd be there, and speaking to completely different audiences and so on, that just taught me so much.
Right. All these other skills like that, sort of working with people and ideas and managing, trying to figure out how to ask for help and how to have people do things for you, or you do things for them, or whatever. Those are all skills. I think those are just as important as learning to program or whatever, if not more. So one last thing I just wanted to mention. A while ago, I asked yourself and Audrey Fan, and then she recruited you to help out with writing this. Maybe you're in the process of writing this book on physiologic MRI or physiologic MRI. How's that going? So it's a book, part of the series of neuroimaging methods and applications. There's a whole series of books, but how's that coming?
Yeah, I mean, I think I have you and Audrey to thank for involving me in the project. I mean, it's been a lot of fun working on the book, and we're starting to get the first few chapter drafts in, and I think I'm also really looking forward to kind of reading them and doing all the editing with Audrey. But I think the thing that I find most interesting about the book, exciting, is really that the physiologic MRI, it's such a broad topic. You have, again, all these things some things we mentioned, but you have all these kind of physiological processes, perfusion, oxygenation, metabolism, and all these kind of things that are usually handled like separately, which is fine because they are kind of processes by themselves, but they're so interrelated. They're just so, you cannot really truly kind of completely separate them from each other. So I think having them all in one place in one book, and also trying to have this kind of to kind of wrap it all into a discussion of brain physiology in general. So not just kind of as it directly relates to imaging, just kind of in general it kind of hopefully will turn out to be something like a book that I wish I had read when I first started working with MRI, because it can get, just also on the most basic level, just in terms of the terminology that different people use, people who are focused on different things and so on. It's so confusing. People use different terms sometimes to mean the same thing, and I hope that hopefully the book will try to clarify those things a little bit.
Yeah, I'm excited to see it come out. I think it will fill a gap in, I haven't seen a book like that, and I think that it would be really useful for the field as well, not only of fMRI, but of a physiologic fMRI, and because, like you said, there's groups that are kind of siloed, all kind of doing different things a little bit, and it'd be nice to have it all in one place so we can sort of catalyze more people working on this. So yeah. So last question, I like to, is there any advice, so far in your young career, but still advice that you might want to give maybe MD PhD students or going into, thinking of research? What are your thoughts on something useful that they could come away with?
I think I'm generally pretty bad at giving advice, but maybe just what to reiterate what I said earlier, like surrounding yourself with people that maybe initially seem like they're doing very different things that maybe aren't super related to what you're doing. I think I was very pleasantly surprised about how much I learned from those interactions, and to just kind of, like you mentioned at the beginning, I'm also not very good at, to be honest, at putting myself out there, but just forcing yourself to do it. Just anecdotally, I when I was thinking about this interview earlier, I just thought of this one time at the very beginning when I started working with fMRI and fMRI-based hemodynamics, and I gave a talk to at least like an internal lab group type talk, but everyone there was like a methods person, and I think I spent like 3 minutes trying to explain how this Baudelaire calculation works. It's a cross correlation, right? Essentially, I didn't know what cross correlation was at that moment. Like, I just did not. And I spent like 3 minutes or something trying to explain how it all works and I created this like animation that shows it and so on. And then someone in the audience just kind of looks back at the audience. I mean, very helpful. I did, but just said, yeah, it's a cross correlation. And that was it. That's all I really needed to say, but I didn't know it. But I learned just from that. It's amazing, I think. And of course, at the beginning, you're like, you're a little bit disheartened and so on. Like, why don't I know this very basic thing that everyone seems to know? But you learn a lot from these kind of interactions. So I think just, try to surround yourself with, as many kind of different people that are working on different things, and you learn a lot.
Yeah, well, that's great advice. Actually, and one thing that struck me is that, you're exactly right. Sometimes people do things and they think they know what it is, but until you actually are forced to interact with someone and explain it, then you really learn it. Then you have to then it really forces you. And a lot of people have blind spots if they don't interact with enough people that force them to communicate well. So
Absolutely.
That's a great learning method. All right, great advice. And this is great. And hopefully it'll be a successful on-ramp to clinical applications someday. And I appreciate all your work that you're doing. And thanks for coming on the podcast.
Thanks so much again for inviting me. It's been a real pleasure.
Thanks.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Xiqian Michelle Li and Ömer Faruk Gülban.